I'm very happy to introduce the second team, finally, of speakers. Mariëlle, who is a clinical researcher, a research collaborator at the Nephrology and Hypertension Service at the CHUV, and who's working mainly with Dr. Grégoire Woersner, who's going to be giving us a little introduction or the background of the talk. I can see you, Grégoire, in the side.
Yes, hello everybody.
Hello. And so this project started in 2015, from what I understood, with a previous, together with the previous CIBM staff, who's now Domenica Buetti, who's now at CISA in Trieste. And so it's a long-standing collaboration. And currently they're working with Andrea Da Costa. And yeah, thank you for your for giving this talk, and I'm looking forward to hearing all the exciting research going on.
Yes, thank you, Pina, for giving us the opportunity to present our work. So I will let Mariëlle talk about science, but I just wanted to introduce why we came to do some fMRI in the brain and link it to blood pressure. And this is the topic of the background. So I'm working in hypertension and nephrology, and you see that it's a worldwide problem with more than 1.13 billion people that have hypertension. And you see worldwide the prevalence is about 22%. It's high in Africa, it's high in the Southeast, it's also high in Europe. And it's even higher as you get older. If you are over 60 years old, 60% of the population has hypertension. So it's really an important problem of public health. And you see that those who have hypertension, of course, they can be treated with medication or activity or diet. But you see, it's only 1 in 5 people that have hypertension under control. And this is a big problem. And as I said, when you have hypertension, the risk is cardiovascular. You can have a stroke, you can have heart attacks, you can have your arteries that get stenosis, all of this. And the first sign is to just measure the blood pressure that says you are at risk of having this cardiovascular complication. But one of the ways to get rid of the hypertension is to treat, and we have good treatment that reduce blood pressure, and these are blocker of the renin-angiotensin system, diuretics, and calcium channel blocker. This is the background, the gold standard of the therapy. You see that you can, on the right part of the screen, that you can combine them and with 3 different medication. And if you still don't have your blood pressure control, you are resistant hypertension. And I will come to this just shortly. So these were the treatment with medication. But in the last 10 years, you see that there were device-based treatment that started to appear, especially renal denervation, where with a catheter, you puncture the femoral artery and you went up to the renal artery, and there you delivered radio frequency to destroy all the sympathetic nerves that are around these renal arteries. There were also baroreflex activation therapy, where you deliver some stimuli to the carotid body, and there's a loop to the brain that goes back to the sympathetic system, and I will come to this just now. There are other, but minor, they are not into clinic, deep brain stimulation that has been tested, which is known to influence blood pressure, also vagal nerve stimulation, median nerve stimulation, but these are very rare. The most advanced device-based treatment are the 3 on top that you can see on this slide here that have been bolded. So what's the role of the sympathetic nervous system? You say we work in nephrology, and there's a crosstalk between the brain, especially the center of blood pressure control, which is in the brainstem. And when your kidney starts dysfunctioning, there's a lot of information going to the brain. And this integration also to higher center after gives a feedback to the sympathetic system, which activates, stimulates renin on the kidney. It does vasoconstriction, and it also reabsorbs sodium in the kidney. So there's a lot of interaction between the sympathetic nervous system and blood pressure, and it's a critical pathway for the initiation and the maintenance of this hypertension. What we knew is what's happened in the kidney. We also knew that these new devices were affecting the sympathetic nervous system, but nobody knows or nobody knew what was happening in the brain, especially the center of blood pressure control that were in the brainstem. So this was our initial question. Is it possible to assess brainstem activity linked to blood pressure control? And if yes, how should we do it? And this was the question we had 5-6 years ago when we started asking questions to radiologists, neurologists, and what came up is you have to use bold MRI. So we said, okay, we use bold MRI. We were very naive with how to assess brain activity. So we went to people who knew how to do it. And we come up to the bold fMRI to use as a technique. But then the second question is linked to blood pressure. What was the best test to do? You see that all the tests that figures on this slide, mental stress or physical stress, they all raise blood pressure. But we chose the one which had the most impact on the blood pressure, which was the cold pressure test. So we had to adapt it for obvious reasons for the MRI. And we were also guided to use the 7 Tesla MRI because we were looking at small structure, which is the brainstem. So once we chose the test, we had to do a pilot study, find also some financing, and we got help from the cardiology and from the hypertensive foundation to start this pilot test. And this pilot test will be presented by Mariëlle. So the question we had was, is it possible to detect the changes in both fMRI signal intensities in the whole brain and the brainstem in response to a modified blood pressure test using the 7 Tesla MRI scanner? So now it's your turn, Mariëlle.
So we designed a small study in which we used ice cold gel packs wrapped around the feet as a stimulus, and we used a simple blanket as a control. Outside of the scanner, we measured the blood pressure during the cold pressure test, and inside the scanner, we stimulated 4 times for 1 minute with either the ice cold gel packs or the blanket. So we had 2 runs with 4 stimulations with the ice cold gel packs and 2 with the control. So here are some technical details in this study. So for structural imaging, we used the MP2 range sequence. We used an isometric voxel size of 1 millimeter. For the functional imaging, we used the 3D EPI sequence with some advanced accelerated bold fMRI techniques with an isometric voxel size of 2 millimeter. And for 1 run, we had 314 whole brain volumes. We also recorded the respiration and the heart rates during the functional runs. For the analysis, we used the SPM12 program on the whole brain, but also we used the brainstem specific analysis. So we reoriented the structural and the functional images. We corrected for motion and co-registered the functional with the structural, normalized them just with the atlas which was in the SPM, and then smooth them. But for the brainstem specific analysis, We isolated the brainstem and the cerebellum using a soot toolbox, and then used a specific normalization for the brainstem, because these are very small structures in the brainstem, so we need to use a different normalization. And then we reslice the functional images and smooth them as well. For the first level analysis, we put them all together in a general linear model with the co-regressors for the movement, the physio, and the scanner drift using the retro-ICOR method. And this was giving a statistical parametric map for either the whole brain or the brainstem specific. And then later on, we all put the subjects all together. So this is the GLM in our study. You can see nicely in the first column the stimulation. We also added a decay because there was still some signaling left after stopping the cold pressor test. We had 6 movement parameters, and there's 9 cardiac and respiratory regressors, and also 3 parameters for the signal drift. So here are the results of the blood pressure measurements, and you can see that during the cold pressor test, there's an increase in the blood pressure. And there's also a slight increase in the stress hormone, norepinephrine. So this is validating our cold pressor test. When you look in the MRI images, we could see an increase in activity in various regions in the brain. So in the supplementary motor area, in the mid-cingulate, and also in the insulae. But we couldn't see anything in the brainstem. And this is using the whole brain method. Looking into the time course of this signaling, you can see there's a nice increase in signaling just after 8 to 10 seconds. And then it's going down again to basal levels in all regions activated in the whole brain. When we use the brainstem specific analysis, we now saw a nice increase in activity in the dorsal medulla region of the brainstem. And this region encompasses the nucleus of the solitary tract and the dorsal motor nucleus of the vagus nerve. And looking into the time course, there is an increase in the signal intensity, but it's less clear than in the activated cortical regions. So based on this small study, we could see that the cold pressor test induced detectable bold intensity changes in the cortical brain regions that are known to be involved in cold stress sensation. And we also could see some in the medulla, but for that we had to use a brainstem specific analysis. So based on this small study, we had some more questions. Does the bold response of the brainstem region to a cold pressor test in hypertensive patients differ from this response in healthy volunteers? And another question we asked ourselves is the bold response to a cold pressor test affected by renal denervation in resistant hypertensive patients? So to answer these questions, we designed a bigger study, a case control study with 3 visits, including a group of hypertensive patients which were off medication, a group of normotensive healthy participants, a group of resistant hypertensive patients, and also renal denervated patients. And in another sub-study, we wanted to measure the bold responses in resistant hypertensive patients before and after the renal denervation. So for the bold MRI in the brain, we made some changes and improvements. So for structural imaging, we wanted to use a smaller voxel size, an isometric voxel size of 0.6, to be able to see also the smaller structures in the brainstem. And also for functional imaging, we lowered the voxel size to 1.5 isometric voxel size. And we also use now a different sequence, a simultaneous multiple slicing sequence. And we added some other functional runs, especially to also be able to measure functional connectivity. So we added a resting state functional imaging. And of course, we had a cold pressor test, but now we are not using anymore the ice cold gel packs, but a bath in which you can put the icy water, which is quite stronger than the gel packs. We also use a control test for the pain. So we use a pinprick, but that we do only on the healthy subjects. And because of the small voxel size, we might have more distortion. So we added a top-up acquisition after each run. And we added also a diffusion-weighted imaging to be able to measure structural connectivity. We also made some changes into the stimulation paradigm, in which we now alternate ice-cold water with body temperature water, and we stimulate them for 40 seconds. So 3 times the cold stress in one run. And for the pain stimulation run, we alternate with a cotton swab. And of course, we record respiration and pulse frequency during the functional runs. So then going to the very preliminary results, we could see when we measure the blood pressure continuously this time outside of the scanner, there's a nice increase of the blood pressure during the cold pressor test, which is quite strong. And also the heart rate increases during the cold pressor test. Whereas during the pain test, the pinprick test, there's no increase in blood pressure nor in the heart rate. And during recovery, the blood pressure is going back to the baseline levels in the cold pressor test. When we look into the MRI data for the whole brain analysis, we could see again this supplementary motor area, the mid-cingula and the insula, that were activated during the cold pressor test. But now we see some other regions as well, like the thalamus, the left caudate, and the right putamen, which might also be involved in the cold sensation. But this is still without applying the distortion correction. So it's not very precise yet. And also in the brainstem analysis, we see some more regions coming up that are activated during the cold pressor test. They're still in the dorsal medulla activation, but also in the pontine region, more in the middle of the brainstem and the higher parts as well. But this is really on a small amount of healthy volunteers. In this study, we encountered several challenges during acquisition. We have the challenges of the quite demanding sequence we use, which created the risk of the scanner crashing. And it might also be a challenge to simultaneously activate the sequence, the MATLAB script for the stimulation input and the recording of the physiology. And also during the processing, pre-processing and analysis, we encountered several challenges. As I already mentioned, the distortion of the functional images due to the lower voxel size. We have effects of the scanner crashes. But a very big challenge is that there is no human brainstem atlas available to identify the structures that we see that are activated during the cold pressor test. And also, as we isolate the brainstem and the cerebellum, how are we gonna do the connectivity analysis together with the whole brain? So how to put it together? And also for us, without a background in MRI imaging or engineering, it's a challenge to use all these different analysis programs. But in future, we want to do as well the structural connectivity and the functional and resting state connectivity to see the connections between the subcortical regions, the brainstem, and the different cortical regions, and compare them in hypertensive patients with the healthy volunteers, both the structural and the functional connectivity. And in the study, there are many people involved, specifically our department in nephrology, but also Sandra, who's helping a lot with the acquisition and as well the data analysis, and several other people in different parts of this study. Thank you all for your attention, and I'm open to answer questions.
Okay, thank you very much, Mariëlle. Great talk. Are there any questions from the audience?
Hello, this is Mary. I have a question. Thank you, Mariëlle, for this very interesting research. Well, actually, two questions. The first one, could you please confirm, so if I understood properly, when you do the whole brain analysis, you don't see activations in the brainstem. When you are just focusing on that area, then you see them. So I hypothesize this is just because at the global brain level, there are registration errors that make any effect disappear in that area, or is that the effects are lower still as compared to the other brain areas?
I think it's mainly due to the normalization. It's in the cortical region, all the structures are bigger. Mm-hmm. So there's the normalization is less fine than in the specific brainstem analysis. Mm-hmm. So you're missing some activation using that normalization.
Okay, thank you. And the other question was about one of your challenges that you are pointing out. You are missing a structural atlas for the brainstem area. I'm not expert in that area, but I was wondering, you are using a very nice sequence, a structural sequence, the MP2H on one side, and you have as well the diffusion in your study. So could you not, from these two sequences, directly segment or split a more subject-specific region that you are interested in? In the MP2H, for instance, you have very nice intensity contrast in some areas like the thalamus, that is partial volume, maybe a little bit like in the brainstem where you have white and gray together. Mm-hmm. It's just a thought. Not sure if it makes sense or not.
I don't understand exactly what you mean.
You're saying you don't have an atlas for the brainstem, but could you just segment directly, so find out the regions of interest from the subject by doing some image processing directly on the using the intensities of the MP2 range?
Just to see the structure.
Yes.
Yeah, but the structures in the brainstem can be really small, like 1 millimeter wide.
Okay.
And very long.
Okay.
So it's really very hard to do it that way, I think.
Okay. Thank you.
And this was a very good question. As you understood from the beginning of our talk, we're quite new in the interpretation of the MRI and bold MRI signals, and this was exactly our chance to be able to present our work, because if you have ideas of how to help us, we are welcoming these ideas of how to analyze and get a better picture of what the brainstem does and how it works in connection with the higher cortical areas. So any question that was asked, we are going to go further inside to see if it can help us analyze the data we have. Of course, we have a lot of data, but we still haven't done a lot of analysis yet, and every good idea is welcome, of course.
There is one group now trying to develop a human brainstem atlas, and there's bits and pieces I can find in literature, but it's not really a whole human brain atlas. So, for instance, the arousal network in the brainstem, that is published already. So it's just these pieces, bits and pieces. I can try to find a little bit what structures it might be that are activated.
Yeah, I can. If I can add something, I agree with Mary that you could use the anatomy information to extract the bold response. You can try, and even staying in a single subject space. I mean, without trying to go into the MNI, you could just see a single subject structure from the MP2 range and extract the bold fMRI response from there for the task, and look into that. So look into if you have some cluster.
Okay.
And also, are you interested, like, because I saw you do, you did like whole brain, but have you thought also to just have this like a small field of view and just do some fMRI on a brainstem? I mean, you could even decrease the spatial resolution even more. But I guess you also need you want to see the whole brain effect, or?
Yeah, we want to see the whole brain, also to be able to do functional connectivity with the cortical regions.
With the rest.
Yeah.
Then also for functional connectivity, you can try to stay on a single subject space and extract from the MP2 range, because you have 0.6 millimeters, I think. So really high resolution for the structure.
Yeah.
And extract bold response from there on single subject, and then do functional connectivity for instance.
Okay, thank you. We will look into that as well, as we are in the beginning of the analysis at the moment.
Perfect. Are there any questions, other questions from the audience on the talk, or? No? Okay. Well, thank you once again, Mariëlle and Grégoire.